So Wenzhong Xiao is Assistant Professor of Bioinformatics at Harvard Medical School and Director of the Inflammation and Metabolism Computational Center at Massachusetts General Hospital. As a world expert in computational genomics, he develops bioinformatic and statistical tools for use in understanding human diseases, especially in studies of immunometabolic response. His talk today is entitled The Severely Ill Patient Study of ME-CFS. So please join me in thanking Wenzhong.
Sure. Yeah, I'd like to thank the organizers for this opportunity to share some of the results that we have from this severely ill patient study that Ron mentioned earlier. As Ron mentioned, all the funding for this study came from the ME-CFS project, which was a grassroots effort by the Open Medicine Foundation to support ME-CFS research. So as we all know here, ME-CFS is a serious long-term illness affecting, unfortunately, multiple organ systems of the body. So this makes the disease harder to understand. So if we see the diagram on the right here, clockwise, you get these muscle systems, the GI tract, the immune systems, the heart vascular systems, and the HPA neurohormonal system that are all affected potentially by the disease, and that lead to the potential problem in the central nervous system that we see from these patients. So to understand this problem, again, as Ron mentioned earlier, we probably have to look at the patients carefully first and see where the signals are, and then design experiments to test our hypothesis. So I think I showed this a couple times before. In terms of the study design, we basically wanted to look at a few severely ill patients, but look at their genes, their genomic sequence, their RNA sequence, and expression level, their proteins, the metabolites, and see what are the differences between the patients and the controls. And further, we would like to check the functions of their cells, for example, the NK cells, the muscle cells, and again, see whether there's a signal between patients and controls. And also, we want to check on the organ functions of each of the organs that we can test through clinical lab tests, and see whether there are particular clue that we can use and correlate with molecular level signal that we can see. And obviously, we know that the microbiomes and, as Ron mentioned earlier, the environmental exposure might also contribute to the disease phenotype. So that was the plan for this study. And there was a question that came up yesterday: how would we actually define those patients as severely ill patients? And to remind us that this was the first study done by the Open Medicine Foundation at Stanford. So the inclusion and exclusion criteria that we picked at that time was that the patients had to be diagnosed by a specialist as a ME-CFS patient, and the patient has to be homebound and spend more than 14 hours in bed, as measured by sensors such as Fitbit and the patient and family report, and also the SF-36 physical function score of the patient has to be less than 70, and the Kanofsky score has also to be 70. I guess if we were to design a study, again, we probably would pick better definition on the severeness of these patients today, but this was what we did a couple of years ago. So as Ron already mentioned, since we tried to look at the characteristics of the patients from the molecular level, the cellular level, the organ level of the patients, and the clinical phenotypes of the patients. This involves a lot of different type of analysis, different technologies were used, and different technologies actually had to be developed. So there's a number of researchers at the Genome Technology Center that actually did the wet lab, and also the clinicians and the clinical researchers who actually recruited the patients and did the sample processing. I just want to acknowledge them first. So our team tried to look at the patient information collected in pretty much three perspectives. One way to look at this would be to consider the patients as one group and the controls as a separate group, and ask the question, what are the differences for each one of the type of the measurements between the patients and the controls. So that's straightforward. And since we measured patients' information at multiple levels, we can then actually look at each one of the patients across different type of measurements and try to correlate the molecular measurements with specific phenotypes of the patient. Obviously, in order to do this, we have to actually define well the phenotypes of the ME/CFS patients. And as I guess a lot of people in this room know, that's actually a challenge for diagnosis of CFS and define the phenotypes of CFS. And lastly, we want to integrate what we learned here with actually what has been done in the past about this disease, as well as other relevant neurological diseases, and try to gain insight about the cause of the disease and the potential treatment. So that's sort of the general plan here. And as Ron mentioned earlier, one of the things that we put as higher priority is to get the data online so the researchers across the world can actually access to this data. So far, there are about 200 researchers having access to the data. This is the website, and I just listed a few here. Obviously, we'll hear from Nell soon about his findings. And David Aspen, who's a researcher at Los Angeles, is looking at the microbiomes and helping us to figure out its impact on neuroprotectant metabolites in the patient. And Dawei Li, who's in Vermont, is using this data to look at the retroviruses in the genome and see whether these endogenous retroviruses actually potentially play a role in the disease. So again, as I mentioned earlier, defining the precise phenotypes of ME/CFS patients is actually a challenge. So this is what we did. So we basically used 4 types of measurements on these patients. I don't think it's perfect, but that's our first try on defining patient phenotypes. So we have the sensor data from Fitbit, and we have the questionnaire data to measure the quality of life, for example, SF-36 or Kanofsky or PROMIS, to compare patients versus controls. And as you can see, there's a clear difference between patients and controls. We should probably have used a better questionnaire, but at that time, that's what we knew what to do. And the cognitive testing showed somewhat a difference, which might be relating to, for example, brain fog in the patients. And as you can see, there's a variance between patients versus controls in terms of their cognitive ability. And as we know from, for example, the 2015 Institute of Medicine report, sleep problem is one of the core symptoms of the disease. And if you compare patients versus controls, you see a clear difference. So this is probably something that everybody knows. The patient is homebound, bedbound. So in terms of number of steps that they took based on sensor data is very low in most of those patients. And this is the quality of life measurements from SF-36. So what we can see immediately is that these are the patients that we used as controls, and this is the general US population measurements. And you can see that the ME/CFS patients that we studied, since they're severely ill, they're far away from the controls. And in the middle, you can see most of the well-known significant human diseases, and they're actually closer to the controls than the severely ill patients that we see. So I think it's clear that ME/CFS patients suffer from debilitating diseases, and that we should study this disease seriously. And as I mentioned earlier, there's significant difference between sleep data in the patients comparing to the controls. So the convention that I use in these slides are the orange ones are always patients, and the blue ones are the controls. And you can see in terms of the REM latency, the stage 3, sleep latency, and the movement arousals, etc., the pulse, there's significant difference between patients and the controls. I just want to bring this up. So Ron mentioned some of the techniques that developed in the Genome Center about detecting viruses or pathogens in the patients, and I just came across this paper or article in the Scientist, which is the latest before I came here, and as you can potentially read here, the title is The Viral Brain, and what it says here is that scientists may need to seriously reconsider the hypothesis that the pathogens can play a part in neurodegenerative diseases, and specifically Alzheimer's, Parkinson's, and some of the other neurodegenerative diseases were mentioned. So the reason I want to bring this up is twofold. One is that there was a question, I guess Rob asked it, what actually caused CFS patient the disease? And I guess, for a lot of the well-known diseases where perhaps orders of magnitude more resources have been put to the research, even today we don't really know what actually caused, for example, Alzheimer's or Parkinson's, let alone for ME/CFS. So I guess our community probably face much bigger challenge with limited resource, and that makes the patient advocacy groups, the foundations, to be critical in our research to actually find the cause of the disease and potentially find a cure. Ron already mentioned this. This was one of the data sets in this study where 20 common DNA viruses were analyzed, and we didn't see a difference between patients and the controls. And this is another view of the viral enrichment study that Ron mentioned earlier. The nanoviruses, another name for it is the transfusion-transmitted virus, or TT viruses. You can see that between patients and the controls, we actually don't see a significant difference. If anything, the patient level is actually lower for this virus comparing to the controls. And we also tested in clinical lab tests the common antibodies between patients and controls, and we didn't see a significant difference either. And the Bartonella and the Lyme tests didn't show a difference as well. However, what we found in the next few slides, I'm going to summarize some of the positive results that we see here, which might lead to new ideas and further testings in a larger patient cohort. So one of the differences that we found was this normal rhyme in in the cortisol levels in the blood, which you would see that it picks up in the morning and get decreased gradually as the day goes on, which you can see here in the controls. We see a flattened pattern in the patients, which might due to the sleep problems of the patient, or maybe it also contributes to the disease. When we look at the cytokines of the patients, we see a significant number of cytokines show difference between patients and the controls, and we compared each one of the differences with what published in the past, since there are a number of publications out there already. For all of them, actually, the direction of the changes, for example, the increased leptin levels, the increased CFS levels, etc., are consistent with the past findings, except that the significance and magnitude of changes are bigger here, presumably because these are severely ill patients, so the signal is expected to be larger. One of the potentially less reported findings is this cytokine brain-derived neurotrophic factor, which is a well-known marker for a number of neurological diseases, for example, severe Alzheimer's. And we see that there's about a threefold decrease of this cytokine in the patients comparing to the controls, suggesting that potentially there's a significant neurological component in this disease comparing to the patient level. So we next looked at the metabolites, and out of the almost 600 metabolites that were measured, about 10% of them show significant difference between patients and controls. And because of time, I only show 3 of them here, which show the biggest difference. And from the, I guess, left to right, or right to left, you get this hydroxyproline, which is known to be significantly enriched in the connective tissues, and the significant increase of the plasma level of this amino acid might correlate or might be the consequence of the protein degradation of the connective tissues, as Nell pointed out. And we see a significant increase of lysines and a lot of different amino acids. Again, that might be a consequence of the increased degradation of proteins in patients that we see comparing to the controls. And we see a significant decrease of this molecule, indopropionate, which is a well-known neuroprotectant molecule. You might know that indopropionate is investigated in, for example, Alzheimer's as a potential treatment or supplement for Alzheimer's patients. So if we compare just the pattern of the changes in the metabolomics data with the other diseases, these are the top ones that, for example, Huntington disease, that are most similar to the pattern that we see in these patients. Again, this probably can generate some of the hypotheses that would require further testing. When we look at the gut microbiome in the patients, as you can see here, these are the patients, these are the controls. I guess one of the striking difference that we see here is these yellow stripes in some of those patients, and these are the Akkermansia species that you see uniquely increased to a large amount in the patients, but not in the controls. And we know, for example, in multiple sclerosis, there's similar findings there. So these are the so-called verrucous microbial species. So next I'll move to whole genome sequencing. Since we only studied a few, because obviously the funding issue, a few severely ill patients, identifying the causal variants in patients comparing to the controls is always a tough task. So we look at this problem through two type of analysis. So the first one is to look for the significantly enriched variants, those variants that at least increased by 5-fold in the patients comparing to the background population. And this is a list of those genes. For example, the neurexin 1 is one of the genes that we're trying to follow up. The CUR genes, which came up yesterday, is another group that we're trying to follow up at the moment. So far, we haven't observed enrichment in variants on the transient receptor potential ion channels, which also came up in some of the previous studies. So this is just an example of the neurexin genes. Just put this here, it's just because it's well known in some of the neurological diseases, for example, OCD, etc. And this gene is more than 1 megabases long in the genome, which make it subject to mutations and other type of variants. And this is basically a plot of each one of the patients, and you can see that there are a number of variants on this gene. Now, to actually pinpoint what's the cause, or if there's a effect on the function of this gene, probably would require larger studies and functional analysis or functional testing. As we already talked about this yesterday, the CUR genes, since they are killer cell immunoglobulin-like receptors on the NK cells, and they work with HLA genes, so it's really well known that they affect NK cell functions, and we see quite significant differences in multiple inhibitory and activating genes in this class of genes. And it occurred to us that actually sequencing the CUR genes well is not a trivial task. And we talked to the IHIW community, which stands for International Histocompatibility and Immunogenetics workshop. We were told that there's actually no well-established protocols to sequence these genes correctly, largely because there's copy number variations together with the variants in the sequence of these genes. So we're developing tools in collaboration with Marcelo at Stanford Blood Center and the IHIW community to try to do this correctly. The other way that we can look at the problem would be to look at those rare pathogenic variants in the patient. So what's shown here, each column is a patient, and each row is the variants occurring on that gene. So it's well known that there's this so-called burden test that you can test the association between rare variants in a disease. So because of time and since we only studied a few patients at the moment, I don't think that we have a definitive answer on some of these findings yet, but some of these things potentially would make sense. For example, as a positive control, you would see this CFTR, which is obviously involved in cystic fibrosis, and most of our, I think all of our patients are Caucasians, so you would expect to see a heterozygous CFTR pathogenic variant in a few percent of the patients. So that's sort of the positive control. And we actually see, for example, this is actually the Parkinson gene that occurs in 3 of the 20 patients. And these are often stop-gain codons, meaning that that one copy of the 2 copies of the protein doesn't work because of a premature stop codon there. And the question then is that whether that actually might contribute to the disease phenotype. Obviously, in the Parkinson field, a homozygous defect of this gene would definitively cause early onset of Parkinson's disease. A heterozygous problem, a pathogenic variant would be significantly associated with early onset Parkinson's disease. And if we study it further, you can see, for example, the ADAMTSL2, which stands for a gene with a long name, but basically it's a metalloprotease gene, and it's known that it's also involved in a series of neurological diseases. For example, there's a paper that just came out, I think this year, which was the largest study of Alzheimer's diseases, where they studied, I think, more than 20,000 patients and 40,000 controls. And this group of metalloproteases are found to be significantly associated with Alzheimer's disease. Obviously, that's huge undertaking of studies, and people still are trying to figure out those rare pathogenic variants for neurological diseases like Alzheimer's. So for CFS, obviously, we need a lot more work in order to get a definitive answer. So one way that we're approaching this problem, so this sort of ongoing research, is to take together the rare pathogenic and enriched variants that I talked about earlier, together with the gene results from previous GWAS and targeted gene studies. There are about 2,500 variants that have been mentioned so far, and then compare that results with the genetic results from other neurological diseases. So this is probably well known. It's the Klimt painting on the tree of life, and if we look at the opposite side, the idea is obviously that the different variants in different genes that might contribute to the disease, and what we actually want to figure out is the common impact or common pathway that contribute to the disease process, and hopefully we can find that soon. So as I mentioned earlier, since we look at this disease quote unquote deeply across multiple levels, one thing that we're able to start looking at is to correlate the disease phenotypes, as I described earlier, with the molecular measurements. In other words, you ask the question, what are the molecular measurements that would be significantly correlated with each one of the phenotypes, and how they would correlate with each other. For example, this is a group of inflammation-related cytokines, as I described earlier, and since they're so correlated with each other, they show a stripe in the middle. And because of time, I'll just show you some of the most significant findings from that, preliminary findings from that analysis. So you get this indopropionate, as I mentioned earlier, that's significantly associated with a number of phenotypes, for example, quality of life, pain scores, etc. And this is one of the microbiome species, which was also shown to be enriched in patients in the publication that Maureen Hansen's group published between twins. And it's also significant here. The cortisol levels, I already talked about it. The PDNF findings, we already talked about it. And the TNF beta is actually a representation of a group of inflammation-related cytokines, which is associated with the outcomes. And we'll get this 5-hydroxylysine. That's sort of the significantly associated with the phenotypes, but on the opposite side. So I want to spend a little bit of time talk about indopropionate, and that's just because it's one of those things that we find to have significant difference between patients and controls in their blood. And it's not only indopropionate, but also we get this phenopropionate, which is the metabolic outcome of phenylalanine. And other molecules such as ferric acid are also lower in patients. So you basically see a pattern of neuroprotective metabolites to be lower in patients comparing to the controls. And this molecule became of interest to us mainly because we know that as human beings, we don't possess the enzyme in our genome that can generate this molecule. So this is tryptophan, for example, and it has to be metabolized by the specific microbe in our gut in order to generate this molecule, indopropionate, which would do its neuroprotective roles in the body as an antioxidant, suppression of inflammation, et cetera, and that's the reason it's studied in diseases as Alzheimer's or diabetes for that matter as well. Okay. So just I mentioned earlier, there's actually only one group of microbiomes that's known to generate this type of molecules, and that's what we actually try to study in collaboration with David Estrin to see whether we can actually identify these microbes and quantitate them. So in terms of next steps, one of the things that we're trying to do is, as I already mentioned earlier, to integrate the findings of CFS patients with findings of other neurological diseases. And the idea behind that is that whether there are common symptoms that can shed light on the potential mechanism of the disease, since we have to borrow strength from somewhere. And the second one is to look at the gut microbiome and try to see whether some of those microbes might be responsible for producing the neuroprotective metabolites that we see lacking in the severely ill patients. I'll skip that, and with the generous support of Open Medicine Foundation, we just started a center at Harvard, to do ME/CFS study. So these are some of the projects that we're starting to do in collaboration with the Stanford Center and with pretty much everybody here in the CFS research community. So, so that include looking at immune systems, and as Ron mentioned, looking at, for example, the RNA viruses, and studying the muscle biopsies of post-exertion malaria patients and see whether there's a clear signal which might be tissue specific, and also look at the cardiopulmonary systems which might contribute to the post-exertion malaise and the orthostatic intolerance of the patients, as well as the molecular imaging of the central nervous system.
Let's thank Wenzhong. Again, we've got time for one or two short questions if anyone has any. Yes, please.
Please.
For those of us that are not familiar with the PPA, and for those at home, could you just elaborate a little bit more on how it's generated? So you mentioned it was generated through the microbiome in the gut. Are there mammalian pathways that can also generate it? And
Yeah, that's a very good question.
And what do you think the targets are in the brain for this metabolite?
Yeah, that's a very good question. So this is actually a paper that came out last year, 2018, and that's, I think, the first paper that actually try to answer the question that you just asked. It's a terrific question, because in the past we didn't know, or we as a research community didn't know actually how phenylalanine, tryptophan, tyrosine, etc., get converted into the neuroprotective molecules in the nervous system. We just don't have any idea. We know that these Clostridium species has to be there in order to generate these molecules, but we don't know the mechanism. So the paper only came out last year to identify the type of enzymes in the microbe that cause this particular reaction. So, that's the first time. And the second part of your question is, what's the protective mechanism of this? I don't know if people have a clear answer to this, but it's the effect of this is well studied, for example, in Alzheimer's and diabetes, and it seems to be lower inflammation and antioxidant.
We've got ourselves back on schedule now, so please join me in thanking Wenzhong for his talk.